Good morning, everybody, and welcome. I'm delighted to introduce my friend and colleague Gonçalo Abecasis as a plenary speaker for this University of Michigan Precision Health Symposium. Gonçalo was born in Portugal, grew up in Macau, and did his work at the University of Oxford in the UK with Bill Cookson and Lon Carden. He joined the faculty here in biostatistics in 2001, was promoted to professor in 2009, and is currently the Felix Moore Collegiate Professor of Biostatistics. He's also a founding co-director of the Precision Health Initiative here at Michigan, and he's currently spending time at Regeneron Pharmaceuticals. Gonçalo is that rare individual who's able to identify the essential problems facing our field and to devise really good solutions. He's made fundamental contributions to many areas of statistics and genetics, notably in family studies, quantitative trait mapping, genome-wide association studies, and sequence analysis. For each of these problems, he's developed rigorous, efficient statistical methods and software tools that are now standard tools used by thousands of investigators worldwide. His work on genotype imputation would take my nomination as the most important advance in statistical genetics in the last 20 years. Gonçalo has a keen interest in the understanding of the genetic basis of health and disease. He's a leader in studies that have identified thousands of loci for macular degeneration, asthma, psoriasis, type 2 diabetes, lipid levels, and other traits, and has played key roles in key resource development projects like the HapMap, 1,000 Genomes, and currently the TopMed project. Gonçalo is an outstanding communicator, as you'll experience soon, a wonderful collaborator, a gifted teacher, and a generous mentor. He was Pew Biomedical Sciences Scholar, a winner of the Overton Prize from the International Society for Computational Biology, and the Kurt Stern Award from the American Society of Human Genetics. He's also a member of the National Academy of Medicine. So please join me in welcoming Gonçalo Abecasis as plenary speaker for this University of Michigan Precision Health Symposium.
Really a pleasure to be here, and thank you for that kind introduction, Mike. And really good to see people from so many parts of the university here in the symposium. I really think that to realize the promise of precision health and to translate the insights we're getting from genetics and health data into really benefits for all of us, it takes many different kinds of people and many different kinds of expertise that extend far beyond just genetics or medicine. And so I'm really glad to see you all engaged and contributing. So the goal of my research has been to use human genetics to advance what we know about human biology. And the sort of really basic place to start in thinking about the possibilities and what human genetics enables is, imagine DNA as an instruction manual, and imagine that each one of us has their own copy, and that we've gone around and made small modifications in each of those manuals. Each one of ours is a little different from everyone else's. And then we can see what happens when we tinker with that set of instructions. Perhaps we'll find that changing a particular instruction results in someone that's a little taller, or someone that's a little more prone to cancer, or all the different kinds of possibilities. As we understand those, take that big parts manual, look at outcomes that we care about, we'll understand biology. Perhaps that will enable us to develop new medicines. Perhaps they'll enable us to just help people understand what's in their future. As I've been engaged in genetic studies, one of the remarkable things that has happened over time is how our ability to collect and organize genetic data has changed. So these are a selection of studies going from about 2000, just before I joined the university, to studies that are still ongoing. And you can see that they've increased in size, both in the number of individuals that are being studied, from about a few hundreds to hundreds of thousands. They've also increased in the level of detail with which we measure DNA, perhaps measuring a couple of dozen genetic variants to now sometimes measuring hundreds of millions of genetic variants. If you combine these two rates of growth, the number of individuals and the amount of detail, the information we get on each one, you'd see that we're collecting about 4 times more data every year or something like that. And that's, quite a bit ahead of the pace of growth of computing power. So that there's one challenge there. I think the other thing that changes is the sort of questions you can ask and the possibilities are very, very different as we progress. In these early genetic studies around 2000, a lot of the work, if you could only measure a handful of genetic variants, was deciding what variants to measure. You'd have a very specific hypothesis about a gene to study and what outcome might be involved, and you design that very precise study. It turns out that we were very bad at making guesses about biology and coming up with hypotheses, and that when you look as a whole, most of the studies from that early period did not replicate. You could not reproduce them if you tried to repeat them. As things progressed, as we started being able to measure thousands and then millions of genetic variants, it became less key to decide what gene should I focus on. You could change the question and say, I'll measure every gene, I'll focus on getting really good quality measurements of my outcome, and then I can just see what the data tells me. No matter what gene turns out to be key for regulating obesity, the data will tell me. This has continued to evolve. And one of the other interesting aspects has been the right ways to use data. So early on, if you were measuring a handful of genetic variants, you could run your study, you could do an analysis, and you could summarize all that you could glean from your piece of data in a research paper, and it would be there in table 1 or table 2, and someone else who wanted to understand how you went about things and your interpretation, or to combine your data with their own data, could go and get your paper and do that. As one started measuring millions of genetic variants, the standard changes a little bit. As you write a paper, you might focus on some particular exciting aspects of the data, write about those, and highlight those in your paper, but someone else could look at your data and study any other gene, ask many different questions. It became clear around this time that it would be very valuable to have not just very complete genetic data, but also very complete health data, because then in the same set of individuals, you could study many different health outcomes. And I think that's one of the early genesis of things like the Michigan Genomics Initiative that many of you are now using in research, where at the moment, as we heard from Sachin, there's data on over 60,000 individuals, and there's both extensive genetic and health data enabling many different researchers and individuals to come and ask different questions. I wanted to take a side tour before coming back to genetics of health to talk a little bit about the computational challenges, because I think there are some interesting lessons there as well. And as I mentioned, genetic data has grown quite a bit faster than compute power. And so one challenge is, can we really keep up? Can we keep using the data well and making good sense of it? There's many problems that one could use as an example. I'm going to take as a very brief example this problem of genotype imputation. And this relies on the idea that since all of us are related to each other, if we had a little bit of genetic data on an individual, but very detailed data where we've measured every position on another set of individuals, we can find little shared bits of chromosome and reconstruct the full DNA sequence of that individual of interest. And this process works remarkably well, and it works better and better as our reference set of individuals grows in size. So here's a little bit of data showing how it's improved over time from a reference panel that was state-of-the-art maybe 3 or 4 years ago to one that was state-of-the-art a couple of years ago to one that's state-of-the-art now. These are genetic variants of different frequency. This is about a frequency of 1%. And it turns out a few years ago we could reconstruct those with about 80% certainty by measuring just a few spots. If you go forward 2 years, now we're reaching that accuracy for variants of frequency about 1 in 1,000, and now down to 1 in 10,000 frequency. That means that something like the average human genome probably has about 3 million differences from another human genome that you'd compare it to. We can reconstruct about 2.99 million of those without measuring them directly with these inexpensive experiments that sample DNA and then trying to reconstruct the rest. So that's all well and good. But here's another part of the challenge. When imputation first was tried out as an idea in about 2007, there were only very few well-characterized samples that could serve as a reference. And doing these analyses for a new sample took about 15 minutes. 15 minutes is pretty reasonable investment of time. As we started to get more complete data, that time rose to about 40 minutes, and then 1,200 minutes. 1,200 minutes for context would be about a day of compute time, and you'd say, no, that's still not bad. 7,800 minutes, so now you're thinking about a week of compute time. And now, in this setting, 33,000 samples in the reference panel, 37 million variants, 63 million minutes is probably closer to 100 years of compute time. You see, maybe there we have a problem. And I just told you that we're expecting to go not just to 30,000 samples, but to hundreds of thousands of samples, and not just 37 million genetic variants, but hundreds of millions of variants. And so this is a common problem in many areas of statistical genetics, whether you're trying to do imputation or to model relatedness between individuals or account for population structure. And I guess one really remarkable thing in the problem of imputation has been that as this challenge has grown, statistical tools and methods have evolved in parallel, and they've really more than kept up with the challenge. So if we take that problem that using the original imputation methods would have taken something like a week of compute time, because the algorithms depended both on the number of genetic markers that we were measuring and the number of haplotype pairs, because every individual has 2 chromosomes. We'd have to compare it to every pairing of known genomes at least. Eventually there's a new class of methods that made this just linear on the number of haplotypes in our reference panel. So from 1 week to less than 10 hours. And it turns out that the early versions of these tools were written by statisticians that were not that good at writing code, and folks who were much more keen on figuring out how to write good code, and this turned out to be colleagues working at 23andMe and trying to keep up with millions of individuals using their consumer genetic tests, took those same algorithms, made a few small tweaks in the code, and I think in the end it was less than 20 changed lines, but they increased efficiency from those 10 hours to half an hour. A new class of methods then became apparent a few years later, and those were guaranteed to have lower complexity, and they changed that 20 minutes to 1 minute. And they were also susceptible to code improvements, and so now you're down to less than 5 seconds. And there's now actually a new class of methods that has constant time, irrespective of the number of samples in your reference panel, and that probably would make this process almost instant. At the moment, this new class performs a little less well than those that take 5 seconds, and so for now we prefer to invest the 5 seconds of compute time. All right, so, and I think that the point there is, possibilities for being creative and discovering new solutions are out there, and they're really more common than we think. And there's so many different problems in genetics where this has happened. And I think for those of you who have a broader view of different fields, you'll say, this is probably the same when you think about image analysis or many other aspects of data. Now, let's go back and focus on one particular disease and just talk a little bit about how things have evolved over time. So this is macular degeneration, and then Sarup, formerly here at Michigan in the ophthalmology department, and Lars, who has been at Michigan, has been out of Michigan and is now back, and Xiao Wei, who's a student who's now a faculty member at UT Southwestern. And they were studying macular degeneration over a period of time. And one of the things that was happening was that they were collecting data on more and more individuals. So this is about 2010 when about 5,000 individuals were studied. 2013, about a little bit over 10,000. And this 2015 study included over 30,000 individuals. One of the things that happens is that, as you increase sample size, you can detect genetic variants with more and more subtle effects. And in each of these graphs, things in green are new compared to previous analysis. Early on, you can discern some patterns. And so, for example, in 2010, what we knew about macular degeneration said, variants around the complement factor H gene, the complement factor I gene, the complement 2 gene, the complement 3 gene, and a few others were important in disease. And you wouldn't have to be really great at biology or a rocket scientist to say, this suggests the complement pathway is important in macular degeneration biology. And at the time, there was kind of an unexpected insight. It was not on top of many people's lists of what would be the causes of macular degeneration. As things expand, we start seeing other kinds of genes and pathways show up. Here's an interesting one where CETP, LIPC, LPL all show up, and that suggests, for example, cholesterol is also important in early disease processes. That was a genome-wide view, but you can zoom in to any little bit of chromosome. Here's a particular region on chromosome 6, a set of genetic variants. They don't overlap any gene, so we think they don't change the protein directly, but that they regulate the function of one of these nearby genes. And in this particular locus, most people who spend a bit of time studying macular degeneration would look at this and say, this is probably pointing us to the VEGF gene. And the reason that they would say that is that it turns out that if you block the function of VEGF, you have basically a therapeutic that's really become quite widespread in the last 10 years, and that's turned a disease that was uncurable. It was and is still the most common cause of blindness in the elderly, but now there's a few different anti-VEGF therapies that basically can stop progression of disease and perhaps even improve vision in some individuals. In other cases, you see the same genetic signal pointing to a set of genes, and we don't have as good clues about biology. And this region on chromosome 22 is an interesting one because as I talk to my colleagues like Anand Sarup, some of them will say, it's obviously the case that this signal is pointing to the SYN3 gene because SYN3 is part of synaptic signaling and photoreceptors, which are the cells that are dying as you go blind with macular degeneration, are neurons. And so this must be the key gene involved. But others will say, the reason those neurons are dying is because the extracellular matrix that supports them is degrading. TIMP3 is a key component of that extracellular matrix, so that must be the key gene. And as you get more and more of them in the room, they'll make good cases for more and more of these genes. And the reason is that, at least for now, we're not very good at interpreting or reading out these non-coding signals. And so, if you're a good storyteller, you can make many different cases for many different genes. A couple of years ago, we tried an experiment, actually with Larzu, where we said, what if we looked in detail at each of these genes, and we focused first on TIMP3 and said, let's look for very severe defects that might remove gene function. And what turned out is that those disruptive defects were much, much more common, and although still very rare, in macular degeneration cases. Here's about 28 out of 16,000 cases versus 1 out of 17,000 controls. And this is basically the analogous experiment using nature and using human beings of trying to generate, for example, mouse knockouts. We're saying if you disrupt this gene, that increases the risk of macular degeneration quite a lot. We also know that subtle changes in the region can increase the risk of macular degeneration a bit. We think this is the causal gene. The equivalent mouse experiment would have been to say, let's generate a knockout mouse, let's let the mouse live till it's 80 and see if it develops macular degeneration. All right, so the really nice thing about it is that as you go through biology and look for different examples of these experiments, you can find again and again some that are very, very convincing examples of where human genetics is pointing to a specific gene and to a very specific mechanism. At the moment, I'm spending most of my time at Regeneron Pharmaceuticals in New York. And in our pipeline, and this is all public information, but we have several targets that we're pursuing and where we've developed drugs. Some of these are out in the market at the moment, others are in development, that are based just on this sort of human genetic evidence of saying, if we knock out this gene, we get hopefully a desirable effect, and let's create a drug that replicates that. Now, I wanted to just say that even when the process works really well, and this is probably a super exceptional case, it takes a long time. So this is probably the shortest example of the whole process happening. And back in 2003, Ellen Hobbs showed that if you have a knockout in PCSK9, you get a really attractive lipid profile. You get very low LDL cholesterol. You appear to be protected from heart disease. This is some of their original data published in 2006. Jonathan Cohen was the first author in New England Journal. And you can see, the whole process, eventually developing an antibody, trying it in the first patient that blocked PCSK9, first phase 2 studies, and then 2015, the first FDA approval for these classes of drugs was, 12 something plus years. And these drugs are out in the market now, but they're not actually widely used anyway. And so, and as I talk, for example, with colleagues at Regeneron, what was the history of each of these drugs where we see, that they work very effectively or that there's very good evidence supporting them? And you can find many examples where the process was not 12 years, but it was 25 years or 30 years. One funny fact about the companies, it was founded in 1988, and their first approved medicine came to market in 2008. So you can see very long lag time between one and the other. Okay, one other very interesting thing is, in addition to getting clues about biology, we can summarize the data in different ways and say, actually, I've measured several million or genetic variants in each individual. I know that some of these affect disease, some have big contributions to risk, some have more modest contributions. What if I try to summarize the risk of each individual? In this particular analysis, we're looking at risk of macular degeneration, and we're summarizing the effects of all the genetic variants that were known as of about 2015. If you look in elderly individuals who are in their late 70s, early 80s, disease has a prevalence of about 5%. And here we're just taking everybody according to their total genetic risk and making a prediction. What do we think when this person gets to be about 80, what's the chance that they'll have macular degeneration? And you can see for many people, the risk is a bit lower. They don't have any of the major genetic risk factors. For the top decile, the risk's about 20%. And then if you divided that further, here's a subset of people where the risk is almost 50-50 for a relatively rare disease. Now, in the case of macular degeneration, at the moment, we don't really have very many preventative treatments. We might advise good, sensible things like, avoid smoking, wear sunglasses if you're out in the sun. Those are both two things that are known to be a little helpful. But beyond that, there's no good thing to do. But in many other diseases, we might have preventative interventions available to us. And one very interesting thing that I think requires a lot of thought and a lot of different people to think about the right ways to use is figuring out, what's the required level of evidence to start communicating this to patients? What are the sort of the right frameworks that we do this to generate the maximum benefit and the least worry or harm or anxiety around it? And really, what is the right trade-off from, the cases where it's most clear that there's a benefit to cases where it's just information about your future health, but not really any intervention or prevention to recommend. I think happily there are some very interesting thought experiments that make this problem a little simpler. And one thought experiment is to think, for example, about conditions where we already do preventative interventions. So if you already intervene preventatively to lower cholesterol when you have a high cholesterol measurement, or when you have a mutation for familial hypercholesterolemia, perhaps one way to think about it, as you think about these much fuzzier genetic risk scores, they don't have any single variant that defines them. They're summarizing the contributions of millions of variants, is to say, what's the cut point that would be equivalent in risk to having very high cholesterol or to having familial hypercholesterolemia? And perhaps if I've already decided that that level of risk I want to intervene, let's maybe think about replicating that there. Okay, one of the big challenges that many of us in the field are engaged at the moment is trying to say, given that it's so interesting to figure out these human knockouts, they give us very specific leads about biology, they give us ideas about new therapies, could we really assign function to every human gene with respect to disease by studying all knockouts? What would this take? You'd need to study very large populations. When I made this slide, I wrote tens of thousands of individuals, but that was obviously a very optimistic underestimate. We would need to figure out how to generate very high quality genetic data for all of them. It turns out that most human knockouts are very unique to the individual that carries them. So it's one of the places where imputation can help a little bit, but doesn't help that much. You really need to go and read out the protein coding sequence of each individual. And then, enable many different people to look at that data and explore it appropriately. So I wanted to show you a couple of vignettes. First, about an experiment where we try to engage populations at scale. Started here at the University of Michigan School of Public Health. And it's an experiment that we called Genes for Good. And the idea was that we would invite individuals to participate in surveys. We would send them a spit kit to collect their DNA. They would allow us to share their data with other researchers, and eventually maybe this would lead to some interesting new findings. Since we were naive and all these happy things, we said, hey, where will we find enough people to engage with? And we decided that a good place to be would be on Facebook, where people already spend a lot of their time. And the result was that we ended up with a study that covered all of the US with participants dispersed pretty much across the entire country, proportional to population. There's actually about twice as many as you'd expect based on population in southeastern Michigan for reasons that, are pretty natural and obvious. And we ended up running the study until about 65,000 people enrolled, about half of those with a DNA sample. One of the things that we did to kind of encourage participation was to say, if you join the study, when we get your DNA sample, we will give you an analysis of your ancestry. And as you fill in different surveys, we will let you compare your results with those of other participants. So for example, here's a summary of a panel comparing alcohol use results for a particular individual. They had about half a drink a day, a little bit less than other participants like them, but more than the average participant. They had a weekly pattern where they drank more on Fridays and Saturdays for whatever reason. It's a common pattern, as it turns out. And overall, the data seems to be quite good, high quality. When we look at, say, standard epidemiological trends, for example, between BMI and diabetes, and compare data from Genes for Good with traditional epidemiological studies, we get very, very similar findings qualitatively. Because we are engaging with individuals in places where we could collect data multiple times, we could actually also collect some types of data that were very hard to get in different ways. And so one of my favorite examples is this one from a summary sleep map of 2016 among all participants. You can faintly see patterns like on Friday and Saturday nights, people sleep a little longer. But you can also then ask questions about, when you go across the entire year, what was the day when the average participant had the least sleep? And you could speculate whether it's New Year's Day or St. Patrick's Day, the first day of summer, many different kinds of speculations. But it turns out that for whatever reason, it's this anonymous second Tuesday in November that's associated with some political events for those. So now, then you can, we've collected survey data. Our participants have answered cumulatively several million surveys with tens of millions of questions. And you can then look at the genetics for any of these. Here's a GWAS of tanning depth, which is just an arbitrary example, but one interesting thing is that these loci mirror very well the same loci that you'd see if you did, for example, using data from the Michigan Genomics Initiative, a GWAS for skin cancer. Okay, so that's one way to engage at scale. Sachin mentioned briefly the Michigan Genomics Initiative. I was very privileged and lucky to work with Chad Brummett years ago when it was starting. And you can see that it has some things in common with Genes for Good, and one of those things is the same graphic artist who did this explanatory pamphlet that we hand out to participants when they join. But there's several bits of interesting history here, and one of the interesting things was when we started, we were using a standard consent recommended by the Michigan IRB that was quite thorough, but it was very long. And one of our colleagues, Nick Stanek at the time, really challenged us to try and get a consent that would be easier for participants to understand and follow, because his research showed that when someone fills in the long standard consent, the 14-page document, and then you follow up with them a few months later and say, hey, thank you for joining our study. Do you know you signed up for these follow-ups? And they would say, what are you talking about? I don't remember that. So he really challenged us to say, can you get it all in one sheet of paper where it's very clear what you're asking, and try and cut out all the extra detail and CYA kind of things that really people end up not reading. And the end result was this pamphlet. And one nice thing that Nick's research also, and Chad's research shows, is that if you use this short consent, if you follow up with individuals later, they're much more likely to remember that both that they joined the study and actually what they signed up for. And actually, because I think as we went through different iterations of this, the IRB said, we would like to be able to use this idea multiple times for multiple different studies. We want to put all the things that we think might cause the most worry in there explicitly, so people are not surprised. And so, so some of the things that are highlighted here, hey, some of the kinds of things we might do with your data, create stem cells that turn into other cells, create cells that live forever, use your materials in research using animals. And so, some of the sort of hot button topics actually that we thought, do we really need to ask for those? Those are not yet technically possible. They said, we'd really prefer if you do, because that makes the future process easier if those become technically possible with the samples you're collecting. And and having this more clear consent changed the consent rate a little bit, but it's more like single-digit %. So it really, I think, is a very great thing. There's an initial analysis from the first 20,000 individuals, that's available here at this site at the School of Public Health, where you can basically look up any disease that's coded that can be derived from ICD codes, and they'll say, this is what we know about the genetics of that disease. These are all the genetic variants. These are the ones that are associated. But then you can explore the data. You can see, this genetic variant, what else is it associated with as I look through the health record. And as I understand it, this is in the process of being updated with the current bigger data set. Okay, so I wanted to also talk a little bit about generating data at scale. And one of my first really large attempts at doing this was in the context of this NHLBI TopMed project. And one very exciting thing there is that we ended up in the end generating data on over 100,000 individuals. This was an initiative by the National Heart, Lung, and Blood Institute to add high-quality genetic data to their preeminent studies. And so these were studies like the Framingham Heart Study, the Women's Health Initiative, a study of Latino children in San Francisco with asthma, a variety of studies that they thought were very high priority for understanding heart, lung, and blood disorders. It was really, for us, one of the first times moving outside our campus compute environment and into the clouds. I think I promised a few people in the audience that I wouldn't use my cheesy comparisons, where I say, we generated 1.6 times 10 to the 16 bases of sequence data, and I say, how big a snowstorm around Ann Arbor, and how deep would you have for that to match the number of snowflakes, or if you're more in a summery mood, how many grains of sand in a beach. So I'll spare you that. But one interesting thing was that, we quickly got to a point where the data was so large, it's currently about 16 petabytes, that any analysis of the entire data set would take, probably many, many months and close to a year. And by the time we would finish it, we would be saying, oh, we already have so much more data, we have to start over again. And we ended up working with Google Cloud and got to a point where we could process each of these genomes in about a minute and keep doing it constantly by having access to 20 to 40,000 cores at a time. And so that was kind of a really kind of transformative experience. The data set and some views of it are available online for browsing. And one of the sorts of interesting questions to ask in deciding whether you want to go through the process of getting access to the fuller data set is to find out if for your particular gene of interest, if we have the sorts of variation, including knockout variation, that might be most informative. So here's PCSK9, one of the poster childs for how genetics can be informative. It's not a very big gene. There's a lot of variants that are missense. They change the protein a little bit, but don't fundamentally disrupt it. And in PCSK9, in the TopMed data set, when this slide was generated, there were 7 different stops, early truncations of the protein or frame shift variants. And I remember sitting with a colleague and saying, what's your favorite gene? I bet we could look it up in the data set, and there will be many examples of knockout carriers, and we could try and look up their phenotypes, understand something about the biology. And they said, KMT2D, it's a methyltransferase, and I looked it up. It's a very long gene, so that was a good start. There's lots of variation there, 496 missense variants. But it turns out 0 stops or frame shifts. It's one of these genes that appears not to tolerate stop or frame shift variation. And I ended up probing my colleague a little bit more and said, what does this gene do? And it's like, oh, we haven't been able to generate a mouse knockout because it's lethal, embryonic lethal in mice. And in humans, it causes Kabuki syndrome, which is a very severe Mendelian disorder. And so obviously there are limitations of the approach. It doesn't work for every gene. Now, I want to spend the last few minutes talking about really trying to bring the two together, having a very well-characterized population with rich genetic data. And the example I wanted to use is this collaboration between Regeneron, a consortium of several different pharmaceutical companies and UK Biobank. And what we're trying to do is, UK Biobank is a study, a longitudinal study for about 500,000 individuals in the UK. We have set out to, certainly by this time next year and probably quite a bit sooner than that, to generate whole exome sequence data for all 500,000 participants. In fact, just the last couple of months, we've publicly released through UK Biobank, and I think many researchers here in the room might be accessing the data for the first 50,000 exome sequence participants. It's a very rich data set. There's about 10,000 items related to health and disease in each of these participants. And, and you can ask different sorts of questions. There is actually an existing genetic data set that's based on arrays and imputation that covers almost all the participants. And it already lets you do pretty rich analysis. So here's an analysis of breast cancer that shows both a summary of what you can discover for common variation, but also to say that, through imputation, you can already learn about some of the rarer variants. Here, variants in CHEK2 and PALB2, BRCA2. These are all places where knocking out a gene increases risk of breast cancer significantly. The data set that was made available in March was exome sequence data for the first 50,000 participants. I don't want to spend too much time on this. There are just characteristics of the participant set, but to show, their average age is about 58, 54% female, a little bit economically deprived, body mass average 26, which is actually relatively healthy compared to a US sample of the same age. And then they've been measured quite extensively, from ECGs, retinal images, not in this slide, but brain images, to then extensive surveys down to, dietary habits and so on. We tried to select this first 50,000 individuals to be those that had more detailed measurements at the time of selection. And so, for example, almost everybody with an ECG in the sample is part of this first 50,000. About half of the individuals with the retinal image are also part of that 50,000, and so on. When we look at the data, we see that at the moment, we have at least one knockout variant. LOF is basically loss of function. It's shorthand in the field for knockout in about 17,000 of the 18 or 19,000 human genes out there. So most genes already have one knockout. And we expect that by the time we're done sequencing everybody, there will be 15,000 genes that have over 50 knockout variants at least. So that's been kind of exciting. And when we look at the data, both in the first 50,000 and then an interim update that's not yet released but will be available later this year with now 150,000 individuals, we see that we can find many of the positive controls that we'd expect. So for example, mutations in MLH1 associated with different kinds of neoplasms, or PKD and chronic kidney disease, titin and cardiomyopathy, really kind of a wide range of examples. And I like to actually show this slide, which is based on the current analysis of 150,000, to just show that as you look through different genes, there's just a very big range of what knocking out a gene could do to your health. From these variants in PKD2 that increase risk of polycystic kidney disease by a factor of about 1,000. So there's, in healthy individuals, there's 140,000 healthy individuals, and there's 24 examples of knockouts in this gene, but there's 85 individuals with polycystic kidney disease, and of those, 15 have a variant in that gene. So a huge enrichment. You went from 20 out of 150,000 to 15 out of 85. And as you go through this spectrum, here's other diseases with odds ratios of over 100, then intermediate odds ratios of 20s and 30s, and then down to things like if you knock out filaggrin, you increase your risk of atopic dermatitis, a relatively common condition, by a factor of 2. Or if you knock out NOS3, you increase your risk of high blood pressure by a factor of 2. And these are kind of selected to show the spectrum, like the range of different things that one could learn from several hundred examples of positive controls in this one data set. And I guess you can imagine that this is well beyond what any single one of us could actually pursue and comprehend fully. There's just too many different interesting genes, too many different interesting diseases to look through. In addition to replicating known things, there's opportunities to discovering truly new biology and truly new signals. One very interesting one for many of my colleagues has been this observation that knockouts in PISO1 increase risk of varicose veins. And I guess PISO1 is kind of an interesting gene because its function is to convert mechanical signals like stress and blood vessel walls into electrical signals. So your body says, hey, something's going on, I should adjust in some way. So, but there's several other new ones. So here's one, MEP knockouts increase bone mineral density, and Col6A1 are associated with corneal diseases and so on. And I wanted to finalize with this, which is a well-known, or not well-known, but perhaps very well-studied gene. This is ApoE. So this is where genetic variants in ApoE greatly increase your risk of Alzheimer's. And Alzheimer's is actually one interesting example of the challenges of using a cohort like UK Biobank to study disease and biology. It turns out that at an average age of 58, and with a set of individuals that's actually moderately healthy compared to the average, there are not many cases of Alzheimer's. And so we can look and see, if we looked at the ApoE4 variant that increases risk of Alzheimer's and looked across the cohort, what things would be associated with those variants. And so this is a selection, just thinned a little bit so it would fit. And you see that ApoE variants are associated with ApoB, CRP, and LDL cholesterol levels. This is known. It's known that in addition to increasing risk of Alzheimer's, ApoE is very strongly associated with blood lipids. But then look at these two signals. Mother has Alzheimer's or dementia. Father has Alzheimer's or dementia. So it turns out that we don't have very many cases of Alzheimer's or dementia directly, but we have many individuals who have a relative with Alzheimer's or dementia. And so we see that there's a 70% higher risk for having an affected relative. Much lower down, here is your own risk of Alzheimer's. It goes up by a factor of about 4, but not, a little bit. But the significance is much lower because the sample size is a couple of hundred individuals as opposed to tens of thousands. But then, take cholesterol-lowering medication. Your red blood cells are a little different. But then another very interesting one, even though this phenotype was only measured in about a third of the sample, it turns out UK Biobank kept track of how long each individual took to fill out their survey. They have a health questionnaire and they track how long did it take you. And if you have these Alzheimer's risk variants, you took longer to complete the survey. Kind of a subtle, unexpected thing. And when we look, what's here on this side are summaries of brain images. So at this time, about 28,000 individuals have been subject to full body MRIs, including brain images. And we just said, hey, how does your brain change if you have this ApoE variants? And you can see that there's many subtle changes, both in gray matter, how many lesions we see in white matter, some changes in the microstructure, and really kind of noticeable changes in brain activation and how much electric signal is in your brain at rest. And so these were sort of unexpected things, but they might lead us to, both good proxies to study Alzheimer risk factors and related biology in relatively healthy young individuals and in sort of a non-invasive manner, and also perhaps in sort of early readouts for potential therapies or interventions. Okay, I think that's where I wanted to leave it, so we have time for a couple of questions. Just say each of these studies includes many different contributors to both organize the samples and the data. So thank you.